vaccine response or protection from infection. So before COVID, most of the vaccination campaign do not provide feedback after vaccination. So it assumes that the protection is universal or in the majority of recipients. However, in the early 2020-21, before COVID vaccine is widely available, we foresee there is a potential need for change in this vaccination practice because people may want to know about their own response after vaccination, whether they are protected or not. And probably at that time, we already think that COVID will bring a new normal to the vaccination practice. We don't know what is the response of the public, so we perform a online survey to tell people that basically they are only 50% of people are protected after vaccination, for example, in the full vaccination program, but we don't provide feedback. So do people want to have their feedback? Do they want to know whether they are protected after a jab? In fact, we are introducing a new concept of personalized vaccination. But in order for this personalized vaccination to work, we need a biomarker that gives a quick answer whether a person has a response to vaccination. And we all know that conventional seroconversion will take 1 month or 2 months to have the antibody response. So we ask the public whether how soon they want to have the response or the assessment, and whether they want to have the feedback. And our survey, majority of people want to have feedback, okay? And it's a little bit surprising that they want it early, 1 week after the vaccination. So it's a very common answer, I think 1 week. So then we look at what kind of biomarker we have in terms of vaccination response. So the conventional seroconversion is the best marker we have. So it measures the antibody production, titer-wise of the antibody, 28 days after the booster dose. However, our survey found that people want it in the first week. So antibody response or seroconversion actually is a late biomarker, so may not fulfill the expectation of the public. So a new early response biomarker is needed. So if we look at the immune response, so this is the seroconversion, production of antibody, and it takes 28 days to reach a peak. But we all know that B cell activation in terms of gene activation is much earlier, which happened within the first week after the injection. So B cell gene expression may be a very good early marker. Now we come to measurement of gene expression in peripheral blood, okay? So what we're measuring most of the time is we call it bulk transcriptome. That means it's measurement of a mixture of cells. So because peripheral blood is a mixture of different white blood cells, for example, we show a granulocyte here and a lymphocyte here. And granulocyte, for example, producing 10 transcripts, and the lymphocyte producing 6. And when we measure, it's 16, okay? 16 is what we get. But we don't know which transcripts come from which cells. I mark it but different color, but each transcript, in fact, they are the same. So we call that is the 16 result is a bulk transcriptome data, which do not have the cell-specific identity or don't have the cell of origin. Although apparently it's so obvious that the bulk transcriptome is not a very good biomarker, but so far all our gene expression data are in fact, particularly for the blood transcriptome, are about data. So most of the study look at the differential gene expression. So using heat map or this forest plot and or volcano plot. So they are all referred to bulk data, and they don't have cell of origin information. The other method is, now we all know that we have another method that can give you a single cell gene expression data, single cell RNA-seq. So it gives you, for example, basically every individual cell, you have a gene expression value, okay? So this cell's expression is 7.3, this cell is 10.0, okay? So each cell, every individual cell, you have a expression data. The Peak4 is very expensive, and it's $100 per sample, and will be very difficult to make it for routine clinical use. On the other hand, what kind of data we prefer? We could use a cell population-based data. For example, it may be good enough if I tell you that all the granulocytes on average produce 10 transcripts, and the lymphocyte produce 6 transcripts on average. So we don't need individual cells data, but as long as we have the cell population average, will be good enough. So we call it single cell type gene expression test. So how could we get this data? The gold standard is to isolate those cell types, the individual cell types, and then do the expression analysis. But cell sorting and isolation is also quite difficult to put in routine use. Therefore, we try to think of a way whether we can have use bioinformatic means to get the cell type information for some genes. So in fact, for some genes, we can get the cell type information from the bulk data. So I classified there's 3 types of genes. Group A, the cell type information you cannot determine from the bulk data, okay? No matter how you do it. Group B will be the cell type specific gene that we well known. So CD19 for B cell. And group C, there's also a retention of the cell type information in the data. Okay, now we look at those 3 groups. So this is example of a group A gene. For example, the bulk transcriptome data is 44, but we know the granulocytes producing 4 transcripts per cell, and then the lymphocytes producing 20 per cell. So there is 6 granulocytes. So they are half and half, basically. So it's very difficult to attribute this 44 transcript to either the granulocyte or the B lymphocyte. So in this kind of cell, the cell of origin information will be very difficult to get, okay, or very inadequate or inaccurate if you want to do it. The other group is those well-known cell-specific genes. Okay, so these genes are only expressed by B cells. So for example, CD19, okay, then all the transcript in the bulk data will be coming from the B cells, okay, for sure. Okay, so there's no problem. But such kind of cell is few, and they are all the B cell reference gene. So it doesn't serve the function of seeing whether the B cell is happy or not, or what is the activation of B cell. So it's just good for using as a reference. Fortunately, we have another group of cell genes that are predominantly produced by lymphocytes. For example, in this case, the lymphocytes produce 20 transcripts, and then there's only, for example, there's 6 granules, each producing 12, okay? So the lymphocyte is producing the majority, over half, of the transcript in the bulk data. So we can quite sure the variation in the bulk data are mostly due to the B lymphocyte. So this is the new group of genes that we call them cell type informative genes. That means we could divide the cell type information with a good degree of certainty, although they are not cell type specific, right? So the granules also produce a little bit, but not overwhelmingly enough to tip the balance, okay? So we also derive a way to tell which gene is cell type informative. That means we can determine the cell type origin from the bulk data by using a pair sample. So when we measure the transcript in the whole blood and also measure the transcript in purified cell of a particular cell type of interest, for example, purified B cell and in the whole blood, and then as long as the B cell sample is 10 times higher than the whole blood, we know that this gene will be predominantly produced by B cell in the peripheral blood sample. And we call them B cell informative genes. So these are the example of these genes which are B cell informative. And for example, we saw that this is TNF SF17, which is actually a B cell activation marker. So which easily that we could use that as a cell type informative gene in a bulk sample. So just to compare what the cell type informative genes on the other side. So we only get the FA transcript per cell type. So it's the transcript of a cell population. So we call, because it can be directly measured now in the peripheral blood, so we call them direct leukocyte subpopulation transcript abundance, direct LSTA. Whereas in the single cell RNA-seq, you get data from individual cells. So this is the difference between the two methods. Okay, so we are, after the method development, we apply this method to look at different data sets of volunteers after full vaccination. We only include data sets that have blood sample taken at day 0, day 7, and day 28. So the day 28 will define who is the responder and non-responder by a 4-fold increase in the titer level. And then the day 7 will be the early marker. We want to see whether we can use the blood transcriptome as a predictor for the seroconversion status. So these are the data set that we pull for analysis. So we use these B cell activation genes. We use 4 of them, and using the TNF SF17 and TXNBC5 as the activation gene, and the other 2 as the reference gene. And firstly, we make sure that we can our method directly gauge the B cell expression of these two genes and be using the gold standard results, which isolate the B cell and see the expression of these two genes compared to our method using the bulk data. So we show that on the x-axis is the isolated B cell from the same sample, so which we could express the target gene TNF SF17 over a housekeeping gene in the isolated B cell and put in the x-axis. On the y-axis is the gene expression determined from the bulk transcriptome data using a ratio of this TNF SF17 over another reference gene, TNF SF13C. So we call this ratio as the direct LSTA results. And this ratio, you can see the y-axis value basically is where give a linear correlation with the isolated B cell expression of the marker gene. So from now on, we can reliably measure this gene expression in peripheral blood without the need of isolating the B cell. So we also use another marker gene. We also show that the same. And then we compare the expression of the biomarker gene of the two groups. On the further away is the non-responder and the responder level 7 days after injection. And we see that the responder has much higher level. We also can follow the change. So the green dot are all the responder. And if we follow the change from day 0 to day 7, most of the responder give an increasing slope, whereas most of the non-responder are flat or actually decline. So both of them can be used. The increase of level or the absolute day 7 level can be used as a biomarker. And we also perform meta-analysis to make sure that our data can be validated or replicated. And actually, this change can be replicated in basically all the data set I've shown. And we also do ROC curve analysis, and the area under curve is 0.85 with a specificity of better than 0.85. So just to recap, by using gene expression of the B cell, we derive an early biomarker of vaccination response, and which could be used to measure in peripheral blood without the need of cell separation. Another application of this assay is to use it as a companion diagnosis or diagnostic for patient selection for tumor immunotherapy. Of course, I know that the audience has a great interest in immuno-oncology, so I also try to demonstrate how this marker can be used as companion diagnostics for selecting patients that is suitable for immunotherapy or cancer vaccine. Other use, of course, we can monitor the change of immunofunction during treatment or use for characterize tumor infiltrating lymphocytes. But I just focus on the use of as a companion diagnostic. So what is companion diagnostics, you know very well. So it's the pre-treatment biomarker that try to differentiate a patient whether he or she is will benefit from a particular therapy. And in the context of immunotherapy, the very typical example is the PD-L1 therapy, which is using companion diagnostics to select patients for this treatment. But could we use the B-cell biomarker as a companion diagnostics? So we work on this data set. So let me introduce you to this data set. So this is a study of 112 prostatic cancer patients who received personalized peptide vaccination. And they have very good subsequent survival data. And also the pre-treatment blood transcriptome data. So we have very good opportunity to apply our biomarker. They follow up this patient for as longer than 900 days, but quite a number of patients died within 1 year of the treatment. Okay, so original paper reported 35 genes that predict immune response. They use the typical bulk data differential gene expression analysis. So this typically generate a long list of genes that are different between the response group and poor responder. The top 2 genes are related to granulocyte function, and haptoglobulin is also there, but the function is not sure. So we analyze the data from the perspective of single cell type informative gene of B cell. So using the ratio that we derive from the vaccination exercise, we also use that B cell activation marker and see whether it's difference between the patient with good response and bad response to the tumor vaccine. So we classify patients with good response for those who survive more than 1 year. And the bulk sample is from PBMC, and we use so they have this TNF RSF17, and we use FCRLA as the reference gene. So it reflects the B cell expression of the marker gene. So this is the bulk data collected before vaccination. So for patients with good prognosis, this time the B cell activation is less intense. So the B cell activation biomarker is less activated among the patient with good prognosis. So suggesting that patients who already mounting a active B cell response to the tumor do not benefit much from vaccination, right? So that's why patient has a high baseline B cell activity, less responsive, or don't survive very well. So in fact, it also reflected in the survival curve analysis. So patient with the low B cell activation has median survival of more than 1,000 days. Okay, more than 1,000 days. But patient with high baseline B cell activation, the median survival is only about 300 days. So this is also a demonstration that our method could be applied in the immuno-oncology context and give surprisingly good results. So just recap, what are the methods that we could use to gather single cell type gene expression. Okay, so the gold standard method is still the cell type sorting and then do the transcription assay. And our method, which is have the beauty of able to apply in the bulk data, which you already collected, or actually you can do it in a peripheral blood, okay, without cell sorting, and could be easily done with a few number of gene marker. You can get a reasonable or kind of meaningful results. And of course, the most intense is and more informative will be the single cell RNA-seq. So just to compare the gold standard method, the cell type sorting is not too expensive, about $100 per sample, but it's labor intensive, okay? It takes time. The single cell RNA-seq, of course, gives you a lot of information, but it's very expensive. It's not used in the routine setting. And our method gives you information in between, and is also fairly priced, not expensive at all. We use this bioinformatics means to find out these genes that we can determine from the bulk data and give you a meaningful biomarker, which could be applied in a wide clinical setting. So we call it a direct method, direct LSTA. And so it is also a method that can be directly from the blood to the results without the need of cell sorting. And we hope that it could be, of course, with its low cost, it could be have a variety of clinical applications. And our long-term vision is that this technology could be applied in routine hospital lab, that just like nowadays we do a complete blood count. So all the data has been published in this paper, and we are supported from the Hong Kong Science Park, and we also joined the Geneva Expo this year and received an award. And I have to thank the whole team that make up of all this preparation and results. So thank you for your attention, and we are very welcome any question or future collaboration. Thank you very much.